Well, welcome everyone and welcome to today's webinar. I am Paola Gamboa, Education and Digital Manager of the Graduate School One Health Emerging Infectious Diseases. Thank you all for joining us in this session in our series, One Health Research in French Amazonia. We are delighted to have you with us today, whether you are joining from the Democratic Republic of the Congo, United States of America, Spain, Italy, Germany, French Guiana, or mainland in France, and whether you are a regular or a new participant. I am pleased to introduce our chair for this session, Professor Loïc Eppelwan. He is a recognized expert in infectiology and epidemiology of infectious and tropical diseases. He is a full professor and hospital practitioner in infectious and tropical diseases in the Infectious and Tropical Diseases Unit at Cayena Hospital, Centre André Rosemont in French Guiana Department. Today, we are particularly honored to welcome our distinguished speaker, Professor Roxane Shaube. In January 28, we will welcome Professor Benoit Detoisy, and extra sessions are being planned. Remember that all our sessions are from 12:30 p.m. to 1:30 p.m. Paris local time. A few practical notes before we begin. The 40 minutes presentation will be in English, but please feel free to ask your questions in English or in French. We encourage your active participation and we look forward for a rich exchange of ideas. When asking your questions, do not hesitate to open your camera and to introduce yourself, giving your name, your status, if you are a researcher, a student, or other, and also to tell us which country you are contacting us from. Without further delay, I will now hand over to Professor Loïc Eppelwan. Thank you and enjoy the webinar.
Thanks again, Paola. Thanks to you. Thanks to Professor Solène Kernis to give us the opportunity to create this very special webinar, One Health Emerging Infectious Diseases webinar specialized in the research in French Guiana, French Amazonia. This is today the third edition of our webinar, and we are very, very, very pleased to present today Dr. Roxane Shaube, which is a physician and also a researcher in public health and in tropical diseases, especially zoonosis. And she's been working for years on lepra and Seignier with a very interesting approach, a real One Health approach, because many people say they make One Health approach, but she is doing really that kind of approach, and with very specific, very original things to say about the research she's leading for more than maybe almost 10 years in French Guiana. So I'm very pleased today to share with you the presentation she's going to give to you, because this is a very interesting talk, and for me it's an example for many further generations of young researchers. Roxane, the talk is yours.
Thank you, Loïc. Thank you, Paola. I'm really pleased that you invited me to this webinar and gave me the opportunity to present our work. So today I'm going to talk about what we're working on the thematic of leprosy, especially in its zoonotic dimension. So after a quick introduction on leprosy and on the leprosy in the Americas, I'm going to talk about the EpiLepre project that we've been doing here in French Guiana since 8 years. And tell you about the main findings of this project and the perspectives and ongoing research that we are currently doing. So just a few words to replace the context. So leprosy is also called Hansen's disease. It's a chronic infectious disease that is very slow progressing and with a very long incubation period from 2 to 20 years. It can go up to 40 years. And it's a disease that have variable susceptibility in the population. So we consider that about 95% of exposed people will not develop the disease. It's a disease that affects skin and that causes peripheral nerve damage, mainly affecting extremities like nose, ears, toes, and hands. So it can cause several sequelae, mutilations, and also stigmatization. So it's a disease that caused and still cause problem to many people. So the clinical picture is variable and is according to the host's immune response. And the diagnosis is based on clinical and histological features. So what is causing leprosy? There are two leprosy bacilli. The first one is Mycobacterium leprae, which has been discovered by Hansen in the 19th century. And another one, Mycobacterium lepromatosis, has been discovered only in 2008. So these two bacilli are called acid-fast bacilli for the histological diagnosis because they take red dye when stained. The main problem with those bacilli is that they are not cultivable in vitro, and in vivo they're only cultivable in mouse foot pads and in 9-banded armadillos. And even in those hosts, its doubling time is extremely low, with 10 to 12 days for doubling time. So this is a big problem because everything we do on leprosy is hampered by this difficulty. It's a bacilli that once was environmental, but has undergone a significant genomic reduction and is now an intracellular bacillus. It has a resistant cellular envelope containing PGL1, which can be used in some tests. And it also has a high genomic stability. So every phylogenetic study that we are going to conduct must take this into account. So the epidemiology of leprosy, so it was once disseminated through various human migrations, and it has undergone a significant decrease in the number of cases since treatment became available in 1980. Currently, we have approximately 200,000 new cases per year worldwide, and it's primarily in tropical regions, especially India, Indonesia, Brazil. Even though there is a treatment and it's freely distributed everywhere, there's still transmission persistence, especially in those countries that I've cited. So what are the modes of transmission of this disease? So we know that the transmission is mainly human to human through inhalations of droplets of nasal and oral secretion. And it occurs mainly when someone has a prolonged direct contact with an untreated infected person. So this is really the main mode of transmission that has been known for a long time. But reservoirs and modes of transmission are poorly understood. So until not so long ago, we thought that the transmission was only between humans, but we found that cases in non-endemic areas that are not linked to a sick person, and happen also to people that have never been in an area with leprosy. So what is the source for these patients? Also, we found that cases from the same household are not necessarily infected by the same strain. So what about exposure to sources outside the household? In 2011, there has been a first proof of reservoir, and the armadillo reservoir, which I will be talking about later. So apart from armadillos, there are also a few primates that have been detected with naturally occurring leprosy, especially chimpanzee and sooty mangabes, and also red squirrels in the British Isles. There are also hypotheses on the environmental reservoir. So it could be that people get infected through contaminated water or soil. But all these reservoirs and modes of transmissions are really poorly understood. So let's focus now on leprosy in the Americas. And why in the Americas? Not only because we're talking here from French Guiana, but also because the main animal reservoir known to date is the armadillo. And armadillos are animals that are only in the Americas, from north to south. So there are 25 species of armadillos, and infected wild armadillos have been found in 2 species. One is the 9-banded armadillo. So I'm talking about 9-banded armadillo species complex because recently it has changed and they have subdivided this big species. And also another one, which is a Phryxus extinctus, another armadillo that is found in Brazil. So there's someone asking a question. No? Okay. So in armadillos, the damage that we can see is similar to humans. So they have skin disease, they have nerve damage. So the armadillo is also used as a model for leprosy, physiopathology, etc. So the problem is that in armadillos, you cannot detect this disease clinically except in very advanced stages. And the problem is that in advanced stages, most of them are already dead. You can detect the bacilli in several tissues, which are liver, spleen, lymph nodes, and skin. And you can perform different tests to detect the disease in armadillos, whether by serology, molecular biology, or histology. On the contrary to humans, where most of the people are naturally immunized versus leprosy, 85% of Dasypus mexicanus, which is the most frequent of the armadillo, are susceptible to the disease. So what about leprosy in the Americas? Is it a zoonosis? So on the map, you can see that there have been a few studies done in the Americas exploring the occurrence of leprosy in wild armadillos. And you can see with the red dots that we found positive armadillos in several places. But it's a reservoir, but is it a zoonosis? Armadillo is a reservoir, but is leprosy a zoonosis? So what about the situation in the US. I'm talking about the US because they had done a lot of studies on this topic and they have the most data on this topic. So in the US, in southern US, you have got this Dasypus mexicanus and only one species of armadillo in the US. And it was always difficult to prove any link between leprosy and armadillos. So the main way of proving that there is transmission between animals and humans is with molecular biology. So it has been done for the first time in 2011. They have compared human and armadillo strains of Mycobacterium leprae in the United States, and they found that almost all armadillos and half of the patients in a same geographical area are infected with one of two unique subtypes of Mycobacterium leprae. So it has been it is now accepted that leprosy is a zoonosis in southern USA, but the modes of transmission and the public health significance are still poorly understood. So we don't know how many patients are infected through armadillo exposure versus other exposure, human or environmental. What about the situation in the Amazon? So in the Amazon, there is a persistent transmission and stabilization of the number of new cases, but it's still high, especially in Brazil. You have regions with variable prevalence in humans and high pair endemic foci with a locales where many people are infected. The other peculiarity of the Amazon region is that we have animal that may be reservoirs and also suspected environmental reservoirs. There is a complexity of transmission mechanism because it's leprosy, but also because of the huge ecological biodiversity we have here in the Amazon. We also have 10 species of armadillos in the Amazon and a high consumption of armadillos by people. So even though it's not allowed to hunt, for example, in Brazil, many people are eating armadillo throughout South America. So what about French Guiana? So I'm focusing now on our little territory. So in French Guiana, the prevalence of leprosy in humans was high until the 1980s, until the treatment was made available. Currently, we have around 10 new cases diagnosed each year in French Guiana, and mainly those new patients come from Brazil. Approximately half of them come from Brazil. Half of them do not report contact with a sick person. So we see that we might have different ways of contaminating in French Guiana as also. Hunting armadillos and consuming armadillos is widespread in French Guiana. It is even legal to hunt, sell, and you can find it in many restaurants if you want. So this was the situation, the portrait of the situation of leprosy in the Americas and in French Guiana. So what have we done here in French Guiana? We began 8 years ago with the EpiLep project. This is a project that aimed to understand reservoirs and modes of transmission of leprosy in French Guiana through a One Health approach to be able to study eco-epidemiological links of leprosy. So we subdivided the project in three chapters. The first one was on humans. So in patients, we aimed to describe the diversity of bacilli, to study the risk factors for leprosy in French Guiana, and also try to establish links with the animal reservoirs. Then we had a second chapter, which was focused on animals and especially armadillos, where we wanted to describe the presence and diversity of bacilli, and also produce data on distribution and density of the reservoir. And the last chapter, the environmental one, we aim to detect bacilli in soil. So let's talk about the human chapter. What have we done? So it was simple studies. I mean, it's not very elaborate because the main difficulties reside in molecular biology. So first we did a cross-sectional study whose objectives were to genotype Mycobacterium leprae strains and to try to see if we had any case with Mycobacterium lepromatosis. Why? Because the diagnostic tests that are used for leprosy, they do not distinguish between those two bacilli. You need to go to molecular biology to be able to distinguish those two bacilli. So we have recruited patients diagnosed between 2006 and 2022 and analyze skin biopsies that were taken during their treatment. So we did DNA extraction and molecular biology test, which is the RLEP quantitative PCR to detect for leprae and our LPM for lepromatosis. And when possible, we try to genotype Mycobacterium leprae. And I say when possible because, as I told you at the beginning, you cannot cultivate Mycobacterium leprae, so you rely on DNA quantity and quality without any other means that compared to what you have in the clinical sample. So we have also done a case control study looking for risk factors of leprosy. So those risk factors were, of course, contacts with cases, contacts with armadillos, but also outdoor activities. So it was a 3.1 matched case control study. So each case had 3 controls matched by age and sex. And the controls were patients seen by dermatologists for another reason than leprosy. So the participants underwent a questionnaire on risk factors and exposure to armadillos, whether by hunting, butchering, preparing the meat, or eating armadillos, and also in direct contact. For example, do you garden in a place where you have seen armadillos? Do you go to the forest and things like that? So what have we found in this human chapter? So we were able to date to analyze 68 skin biopsies from 50 patients. So they underwent molecular biology analysis. So we found that some were RLEP positive, but none were LPM positive. So to date, we have no patients with Mycobacterium lepromatosis, only leprae. We have been able to do a whole genome sequencing for 9 patients. So it's not a lot, but it's really a performance, as I told you before. So we have several different genotypes, 1D1, 4O, 4N, 4P, and those genotypes are the same that you can find in other places, Brazil, West Africa, and it's relevant with the history of French Guiana and of human migration. The case control study. So what have we found? So this is a intermediate analysis because it's not complete yet. But we can already see that a lot of cases, but also of controls, are exposed to armadillo. So 65% of cases and 45% of controls do have direct contact with armadillos or do eat armadillos. And so when doing the analysis, we found that a low level of education is a risk factor that is already known in the literature. The fact of having leprosy cases in the family is a risk factor that is known. So it's good that we found it also in our study, but also that direct contact with armadillos is also a significant risk factor. So this is interesting. It's an odds ratio that we found in other studies too. So armadillo consumption, so only eating the meat, was not a risk factor in our study. So what can we say already on this human chapter? So we found no lepromatosis. The strains that we found of M. leprae are consistent with the history of our territory. We found a significant direct exposure to armadillos similar to levels found in Brazil. And even though we have difficulties in the epidemiological analysis of leprosy risk factors because of this incubation period that is so long and other factors, we have found a significant risk of direct contact with armadillos in our population. It's consistent with one meta-analysis done by DEPS. I will give you the reference at the end of the lecture. What we didn't find is a clear role of environmental exposure. And another thing that I didn't talk about yet is that we have done a little study, side study on illegal gold miners in French Guiana that represent a high percentage of our new cases. And we have done serologies on these on this population, and we found a very high exposition level of this population to leprosy bacilli. So let's talk now about armadillos. In French Guiana, we have 4 armadillo species. So here are the 4 species. Those are fantastic animals. I love them so much. So the 2 first armadillo species, main species, are the Dasypus guianensis and capleri. So these are the most widespread, and these are the ones that are allowed to hunting. The other 2 species are protected and much less frequent. As you can see, the 2 Dasypus are restrained to the Guiana Shield area, but the other 2 species are more widespread in the Amazon. Our sample collection. So we have been able to use an ensign collection of serum and skin biopsies that were done in the 90s in the Petit Dam region. But we have also collected samples through a dedicated sample harvesting. We took roadkills, we contacted hunters and restaurants and also the general public to find samples in whole French Guiana. So we used a poster calling for our samples and it worked quite well. And so we were able to collect many armadillos. When possible, we took blood samples, ear, liver, spleen, and lymph node samples. The lab analysis on these samples were were complete. I mean, we did serologies when possible, 2 different serologies. We did histology, like for clinical samples, and we did molecular biology analysis. It's the same analysis compared to human clinical samples. It works very well. And when possible, we try to do whole genome sequencing as well with the same technique as for humans. We also tested 2 different DNA extraction methods to really have the most complete results possible on this collection. What did we found? So we found that on the serum collection, the inside serum collection, we found only 6.6% of Dasypus guianensis that were positive in serology. It was already interesting because nothing had been done in French Guiana nor the Guiana Shield before. So these were the first results. But then the most interesting results were on the dedicated collection. So we were able to analyze 186 armadillos. And out of those, 21.5% had a positive test in molecular biology. We were able to confirm this in histology in almost all of them. And then we also had 38% that were positive in serology. So in armadillos, serology means infection. We found no lepromatosis either. So no lepromatosis in humans and no lepromatosis in armadillos in French Guiana to date. We were able to collect samples from the whole territory, even though we have, of course, much more samples close to the cities. So we have positive armadillos in 3 different species. So that is really interesting. And so on this map, you can see all the blue and red dots are our samples, and red dots are infected animals. So the most interesting thing is whole genome sequencing, because at the end, that's the result we can use to prove transmission from between animals and humans. But we are able to do only 1 strain typing of M. mycobacterium leprae in 1 armadillo. It's still the same problem concerning the fact that we cannot cultivate this bacilli. So we found 1 4P strain in 1 armadillo, and we were able not to do whole genome sequencing, but subtyping of the same strain in another one in the same region. So what did we found is in this chapter is an evidence of a major Mycobacterium leprae in zootic in approximately 1/4 of armadillos in French Guiana. This level of prevalence is it's high, but in almost all studies that have been done in armadillos, the prevalence is quite high. So it's consistent with other studies done elsewhere. So we also found 3 new species of naturally infected armadillos because they have never been tested before. And what we found, even though it's only one animal that we were able to perform a whole genome sequencing on, it is the first non-human strain genotyped in South America. And it's the first time that we describe a 4P strain in non-humans. We also have found a little one that was a very small 2 weeks old animal that was infected. And so that means that vertical transmission is also possible in armadillos. So let's go now to the last chapter. So that's the environmental chapter. So it's the most difficult. It's the one where we didn't progress much yet. And it's also the one that we're going to work on in the future a lot.
Roxane, there are 5 minutes left.
Okay, no problem. Trying to go quick. So Mycobacterium leprae is excreted from upper airways, skin lesions, and feces. It can be detected, the DNA, in soil and water. It has been done in India, in Brazil, and Suriname, this detection. But it's even more difficult in soil and water compared to human or animal samples. So what we know is that the bacilli thrives in soil, in humid and dark conditions. And also there is a possibility that amoebas play a potential role of protecting Mycobacterium leprae through internalization. So we have done soil sampling in a former leprosarium. It's the 6th on the map, and in armadillo burrows in several other locations that you can see on the map in red. So we have taken soil samples, extract DNA, perform molecular biology analysis. And we also tried to do preliminary soil characterization studies, so physicochemical, and also looking for amebozoan communities. So the problem is that even though we changed for techniques, we didn't find, at first, we didn't find many positive samples. In fact, we found only 1 week positive samples in the place where we also had many armadillos that were positive. And then we changed the techniques for a 3rd time, and we found more positive signals in armadillo burrows. We also found that we have a acid soil and a clay soil. So all this is interesting because it can hamper or favorize the bacilli survival and things like that. So we have found some leprae DNA detection in burrows, but we have not been able to do any sequence so far. So we still need to confirm these findings. We also found that amebozoans that could serve as hosts for Mycobacterium leprae are present in soil. So this is not new, but we chose to check this. So now the question is are the detected bacilli viable? So let's move to the main findings of the study. So this is the first study with a comprehensive approach to leprosy. So we combine 3 aspects of the problem, and this is quite new. So we found an importance and diversity of the animal reservoir of Mycobacterium leprae in the Amazon, as I told you. And so we found this 4P strain in an armadillo that we also found in a human sample, but it's not exactly the same strain. So that doesn't mean that there is no transmission, but we couldn't prove prove it yet. So the problem is now, what is the proportion of human cases resulting from zoonotic or environmental exposure in French Guiana? These are the reservoirs and transmission routes hypothesis that we have made on this subject. So we think that armadillos are the main source of bacilli in the environment in French Guiana. And we also think that armadillo burrows could work as a micro ecosystem that concentrates and preserves bacilli. So I think I didn't tell you, but armadillos are digging creatures and they spend a lot of time in their burrows. So the implications are that, well, controlling an animal reservoir, wild animal reservoir, is really difficult. We have no vaccine, and I don't think it's possible, at least here in French Guiana. So we should look for human protection instead. So try to reduce their exposure to armadillos. We also know that tuberculosis vaccination does play a limited role in protecting humans from leprosy. Awareness raising would be also interesting to be sure that exposed population are aware of the risk. So the perspectives and ongoing research on this topic here in French Guiana. So we plan to measure exposition level through serology in the general population and in exposed population. So that could help us calculate the zoonotic potential. We also want to explore further the relationship between the bacilli and the environment, especially soil. The problem is we need to go in armadillo burrows. So how do we go in this very small and complex structure? We have built a robot that is dedicated to this. So we have this wonderful little Armabot for armadillo robot that is still under development, and that will allow us to explore and take samples in armadillo burrows. So it will be soil samples, feces samples, and also arthropod samples, because there is a potential role of vectors that are present in burrows, for example, ticks and triatominae and amebozoans. So here are a few main references that you can look up if you are interested in the subject. And I would like to thank all collaborators, partners, and financiers involved. So this is a topic which needs a lot of collaborators. So people that are working on animal, on molecular biology, robotics, many things, many interesting partnerships were developed thanks to this subject. So it's finished. Thank you for your attention, and I'm happy to answer any questions you may have.
Thank you so much, Roxane. Is there any question from the public? I can see that many people send some applause, hearts.
Thank you.
Camille is raising her hand. Camille, can you please open the camera and pose your question?
Yes. Can you see me? Hello, this is Camilla from Tropical Institute in Munich. Thank you very much, Roxane, for this fantastic talk. If I was young, I would immediately apply for a research project in your area. Fantastic. I've got two questions. One is, why the armadillo? I think I once read that this had to do with their body temperature that was relatively low. Is that true, or is there another reason? And the second question is, you mentioned that one of the strains of Mycobacterium leprae was in fact of West African origin. So I'd be curious to know how closely related they are, and if this tells us about the history of the spread. Was it introduced by slave trade, or is this much older? So did it come to South America when it was first settled by people entering from Asia, actually?
So yes. So for Mycobacterium leprae, the current hypothesis is the spread of different strains to human migration, and especially in the Americas. For Mycobacterium leprae, currently we think that it has been introduced by European settlers and slave trade, and later maybe other migrations too. It's not the case for Mycobacterium lepromatosis, though. So a very recent publication in Science, so I have put you the reference to, found that that was not the case for lepromatosis. That was present in the Americas before Columbus. So what was your first question again? Sorry.
Interesting then to if it was brought in by humans, how the armadillo actually got infected. So was there such a close relationship, or what do we think happened? The second question was, why the armadillo?
Yeah. So we think now that the introduction to armadillos were through humans. So it was quite widespread in humans before the treatment, and we know that armadillos are close to human dwellings, so they could have been infected while foraging in human litter, for example. Yes, so why armadillos? So it's true that this bacilli likes cooler temperatures. So armadillo have a cooler internal temperature compared to many other mammals. So that's true that in humans, why do we found the bacilli in extremities? Because it's cooler, and the whole armadillo is cooler. Another possibility is, of course, the immune system of armadillos, but it's not the only reason, because, as I told you, we have found bacilli in red squirrels which have a high temperature, I think, and also in chimpanzees and other primates. So who knows what other animals are also infected that we don't know about. The problem is that you have to check, you have to want to search it, and then you have to be able to search it, because it's so difficult to just prove the presence of the bacilli in any animal or matrix. Are there any other questions?
Another question? There is a question on the chat from Anna Brittle. Thank you for your presentation. I am a PhD student in France. Is it possible to study leprosy in vivo in NHP models, as there are reservoirs, or are armadillos and mouse footpads models for these diseases?
So what are NHP models, please? Because that I don't know. Apart from that, so
non-human primates.
So there, I'm not really familiar with this topic, but there have been a lot of tests done for modeling leprosy in animals long before our studies, our current studies, and it didn't work in non-human primates. It didn't work well. So that's why at a time researchers tried any animal and they have inoculated leprosy to so many different species just to find a model. And that's when they found armadillos were highly susceptible to inoculation. And it's interesting because at first, so armadillos were used as animal models, and 10 or 15 years after, they found wild armadillos infected. So at first, people were accusing researchers of having spread the leprosy in armadillos in the wild, but it was not the case. It's that they were already infected. So that's interesting historical point. So armadillos are and mouse foot pads models are the current models for this disease. So they use it for physiopathology, for treatment and vaccine tests, and also to multiplicate bacilli to send to labs. So these are the current models. Any other question?
None of them. I saw that many persons connected from several continents. Okay, so Roxane, thank you so much for this wonderful talk. I hope that that will be an inspiration for all the young students and researchers who have joined us today. And yeah, many things are possible today in this One Health approach, studying animals, environment, and not only making retrospective studies of in-hospital patients. So thank you so much to everyone. Thank you, Paola. And thank you so much, Roxane. That was very interesting.
Thank you. Just a few words, if I may. So I would like to thank you for inviting me to present this this work that we've done, and to say to everyone listening that I really discovered this world of One Health approach and transdisciplinary approach, which are hugely interesting. And you may, if you're working on this area, you may need to educate yourself on so many topics, and it's highly interesting, but you need to keep very humble because it's a topic where you see that you know nothing and rely on partners of different disciplines to be able to go forward in discovering new things.
Before leaving, Roxane, there is one last question in the chat in French.
So yes, is there a mouse model with Lepremurium? It was there has been a mouse model 30 years ago. Is it still useful? To be honest, I don't know. I won't be able to answer these questions. I'm sorry. I would need to look it up. Sorry. Thank you very much for inviting me.
Well, she answered.
Thank you so much to all, and the next edition of our webinar will take place in 1 month. Maybe Paula will remind us the good date. And this time, this will be a talk from Dr. Benoît de Thoisy from the Institut Pasteur in French Guiana, and we will talk about vampire bats and rabies. So something exciting from the Hollywood films, Hollywood movies, and different thing also about Amazonia and South America. So we will advise you for the next steps, the next edition. Thank you so much to everyone.
Just to remind you, the next date is January the 28th, so please stay tuned. And thank you for attending this webinar session. Thank you so much.
Thank you. Have a good day and a good afternoon for people in mainland France.